organisms at the prokaryotic and eukaryotic level. This thing which I'm talking about is, I'll come back to this table after introducing how do I remove this part, because this is covering up the topic here. Any suggestion, please? Can I remove this bar from this screen? Anyone? Okay, though I do not know how to do that, but I'll read the heading from here. This is the C value paradox I'm talking about. The heading is C value paradox. Maybe I have it in some other slide also. No, everywhere the topic is covered up. Doesn't matter. So why I'm talking about C value is because C value is the total genome present in a haploid or the haploid genome of an organism. So that is called the C value. And why it is a paradox? Because generally what we believe is smaller the organism, it will have a smaller genome. As the complexity of the organisms increases, so the genome size will also increase. So that means that the genome size should be proportional to the complexity of the organism. But to the surprise of the scientists who were sequencing the genomes and who know the enormity of the organisms, they came to a very startling finding that some of these organisms, which are very, very small, they have some quite bigger genome. And it was like you can say contrary to their expectations. And sometimes these small organisms have a genome which is much, much larger than the genome of some size-wise big organisms. So there is a variation in that. So that is the paradox. So in general, everybody believes that as the complexity of the organism, or let's say to be very layman language, let's say the size of the organism increases and the position of the organism as per the hierarchy in the plant kingdom or animal kingdoms, it goes to a complex or higher level, the genome should also become larger. But it is not found always like that. So that is the C value paradox. And why this is there, for that we need to understand one term which is called the repetitive DNA content. Some organisms which have huge genomes, but they may have less number of genes. So the reason for that is that these organisms may be having a large proportion of repetitive DNA contents in their genomes. And we'll come to now one of the tables which I showed you here. So here we have three tables, three columns: genome size, the number of genes, and gene density. So when we are understanding genome, we need to understand these three terms, and we should also understand what is the correlation between these. So these are the organism, and you'll see that we are from this to this level, we are going towards the higher level of complexity with respect to their organization. So here we have the prokaryotes, we have eukaryotes. In eukaryotes, you have fungi, protozoans, vertebrates, and plants. So the complexity is varying, and you will see that the genome size is also varying. So here in the prokaryotes, you have some genome sizes which are very, very small, 0.5 megabase to 6.7 megabases. Whereas when we are talking of eukaryotes, you can see the eukaryotes, they have huge genome size in comparison to the prokaryotes. And within the eukaryotes also, we know that the lower eukaryotes, they have smaller size, genome size in comparison to the higher ones. So here in these, even in plants, we'll see there is a lot of variation. Amongst vertebrates also, you have some few hundred megabase genome size to thousands of megabase genome size. Plants also, you have just 120 megabase genome size for Arabidopsis, one of the model organisms in the plant groups. And when we are talking about other plants, you can see that it goes even into the thousands and lakhs. So this is the enormity and variation, diversity present in the genome size when we look across the living organisms. And here, there in the second column, there are number of genes. So tentative number of genes, they may be approximate, and maybe there are some more additions to the number of genes, but generally the you can say approximation is almost this number. So here you have small number of genes, and you see gradually the number of genes is increasing. So there is an increase in number of genes, there is an increase in the size of the genomes, and within a particular group also there is variation. So these are few points which we should keep in mind. Then now come to the gene density. So here you see that here is an inverse relationship. Here we have number of genes are less, the gene density is more. Here the number of genes are more and the gene density is less. So this is just like if you compare two cities, like or two living places, one place of two of almost same sizes, or let's say one is big in size and one is small in size. Let's talk of two states like Uttar Pradesh, and let us talk about a small state like Delhi. Let us call Delhi as a state. So these two, in Delhi, we expect the size is small, and in Uttar Pradesh size is big. So, but the number of individuals may or may not be. Again, I'm going hypothetical. The total number of people living in Uttar Pradesh may be spread because the area is huge, so the population is spread. So the density per square meter may be lesser than what is there in the smaller place like Delhi, where per square meter the number of individuals living are more. So that is the case here, that prokaryotes, though they have a smaller genome size, whatever number of genes they have, they are tightly packed. So the gene density is higher even for smaller genomes and less number of genes here. Whereas in case of eukaryotes, you see here the number of genes run into thousands, but the genome, the gene density is comparatively quite less when we compare it even with the prokaryotes. This is because they have enormous size of genome. The gene numbers are also more, but when we compare to that, their space between two adjacent genes may be more. They are not very tightly packed, so the gene density is lower in comparison to this. So there should be no confusion between here. Density means we are not talking about how many genes are there. We are just talking about like in a particular stretch of a DNA, अगर एक same length का DNA आप लेते हो, उस DNA पे कितने genes हैं? Prokaryote पे क्योंकि genome size कम है, तो more number of genes they occupy their positions and they are tightly fitted in the space, so they have a lesser or smaller gene density. Whereas in eukaryotes, we have huge genomes and the genes, they are spaced at distance, because of which they have a lower gene density. So now we have understood of two things, that the genomes of prokaryotes, eukaryotes are different. Prokaryotes generally have smaller genome, eukaryotes have a larger genome, but prokaryotes, they have small gene density, they have a higher gene density. Eukaryotes, they have a smaller gene density. So these, with this thing, it will be easier for us to understand few things that I am going to talk about that, because here when I was talking about repetitive sequences, so we'll talk that to understand like why some of the organisms here, they have huge genomes but the number of genes are less. So what is there? Is the gene size bigger? Say for example here, when we are talking about there's another table, let me talk about that. See here we have the sizes of the genome, the gene numbers. So here we have the organisms with the size, and here we have the gene numbers, and here we have the average number of genes per 1 lakh basis. So when we just have a glance over this, then we can see that when here we are talking all about, see here we have a Drosophila. Drosophila have 13,000 genes, a very small organism, but quite a large number of genes. Homo sapiens are comparatively huge when we compare it with a fruit fly, and they have 25,000 genes, and the genomes definitely will vary. And now let us again compare the Arabidopsis. which has 25,000 gene, which is again a plant, which is very, very much smaller than the Homo sapiens. So it is not always that the size of the organism will decide the complexity of the organism or the total number of genes that are required to perform all the functions in the living organisms. So there will be differences, variations within the group, within the species and genera as such. And sometimes the organisms which have comparable genome size, they may have variation in the gene density, and that may be because of presence of large stretches of DNA which are repetitive DNA, which we do not know whether they are genes or not. So we consider them to be non-functional. And then when we talk of eukaryotes, then there is another thing which I have already mentioned, that is the presence of introns. So I'll come to that also. So when we are talking about a repetitive DNA, which makes the genome size large, even with less number of genes. So sometimes the huge sizes are because of more repetitive DNA. So here the repetitive DNA may be classified under various categories. Again, I will not go into detail of these, but some of the names which you should understand, that these are the ones where the DNA is repetitive. Repetitive means some of the sequences are repeated. Say for example, CA, or there may be it's a dinucleotide repeat. There may be a particular sequence CAUG like that, and that may be repeated a number of times. So that way, that is what we call as repetitive. Sometimes even it may not be a 3 or 4, it may be of 20s or hundreds of nucleotide bases which are repeated within the same chromosome, or maybe in some other chromosome. So when in the entirety of the genome, when we are looking at, then we may find that similar sequences are present at several places. So there are some reasons for the presence of these repetitive DNA, and one major explanation which we can give for the presence of these repetitive DNA is the evolution. That during the course of evolution, several forces are acting on the genome, which may sometimes result into duplication of the things or some of the genomes. Then there is a term called transposition, जो transposons होते हैं, जो See here, this is one gene of a E. coli, this genome of a E. coli. Then we have Saccharomyces, which is a fungus. We have Drosophila and humans. So what here, in general, they are comparing them. And one thing that we need to see is the green part is the gene. So you see that in humans, if we are taking approximately these many number of base pairs, you see that there is hardly, or they are seeing two genes in this particular stretch. This one, and here you have another one. But when we are talking about the same stretch of DNA here in Drosophila, there are more number of genes. So they have numbered it 9. So there are 9 such genes which are presenting the same length of the genome. For Saccharomyces, it is 31, and for E. coli, you can say 57. And here you see that the thing which are making the genomes higher, what are the other things which are occupying the entire stretch of the DNA in case of humans or wherever you have some gaps or less number of genes. These are the introns, which are present or characteristics of a eukaryotic gene. Within the genes, there are some sequences which are removed when final mRNA is synthesized, which will be translated. So these removed regions are the introns, and the regions of a gene which are part of the mRNA which is translated are called exons. So these introns are the extra parts which are present in the genome of a eukaryote. Along with that, you have repeat sequences, and we have some other intergenetic sequences which have not been given some specific name. So we have these intergenetic sequences present in almost all organisms, but the size is expanding when we go higher up in the hierarchy with respect to the complexity of the organisms. Okay. So I'll not go much more deeper than that. We have already talked about. So when we are talking of prokaryotes, the repetitive DNA percentage is even less than 1%. But here in eukaryotes, the percentage, in humans, let's say, approximately 50% of the genome is said to be repetitive. So that is the reason of the higher gene density in the prokaryotes. And because this genome size is low, so prokaryotes are wise enough to utilize the maximum of the given genome. And one such, you can say, modification or optimization of the genome is the presence of polycistrons. In eukaryotes, when you'll see a genome, you'll see that there are some regulatory sequence present before the gene, which will help in the transcription and translation, and towards the end of the gene, which will help in the termination of the transcription. So there are for one gene, there are some regulatory sequences. Whereas in case of prokaryote, you have the regulatory sequences, then you have the gene, and then you have the second gene, third gene, fourth gene, like that. So for a particular function, if there is a requirement of 3 protein or 3 enzymes for a particular reaction which are to be utilized simultaneously. So in case of prokaryotes, all these 3 are linked to some common regulatory sequences, and they are transcribed and translated together. So this way, that extra space required for 3 sets of regulatory sequences is now not required. So one set of the regulatory sequences can take care of the transcription and translation of 3 different genes. As in the case of lac-operon, you must have heard that, which is the operon or the enzymes which are required for the metabolism of lactose. Similarly, we have many other operons. So operons are all these, you can say, all the genes, the regulatory sequences which are lined up back to back, which can take care of one particular metabolic function of the bacteria. And they contain of one origin of replication, whereas in eukaryotes you have different chromosomes, and there you may see that when you are talking about the replication, there are several bubbles, or the replication process can start from at several positions. It's not only single ori or origin of replication as is present in the prokaryotic genomes. This is what I was talking about, the minimization of the, or you can say the best utilization of space in case of a prokaryote. So you have the regulatory sequences here, and you have gene 1, 2, and 3. So these regulatory sequences will switch on the expression. Expression means, when I'm saying expression, that means the DNA here, which has the genes, they are transcribed and sometimes translated. So now the gene 1, 2, and 3, they are all transcribed and the protein products are formed. Instead of you have from here to here attached to gene 1, then you have the same similar kind of things or sequences present again before gene 2 and gene 3. So instead of having 3 times this area, only one representation of this is enough to take care of the expression of these 3 genes. So that is how they are utilizing the minimal space or minimal genome present for all the activities present in a prokaryote. Otherwise also, prokaryotes are small organisms. They are single celled. Their basic necessity or the kind of functions they are doing are comparatively lesser in comparison to the complex organisms. So to sum up, and before entering into the organization of the genome, so I'll just sum up that the genome, it has coding sequences, non-coding sequences. This part comes when we are talking about basically the eukaryotes. In the coding regions, we have genes, which will be translated or transcribed and translated to form some proteins. But then in the coding region within the genes, we have some other sequences which may be introns. UTRs are the untranslated regions which are not translated but which are required for the beginning or you can say initiation and termination of the transcription process. In the non-coding region, we have intergenic sequences, which may further be classified into microsatellites, macrosatellites. Then there are some unique sequences. Now, these unique sequences are sometimes non-repetitive. Some of the genes which becomes non-functional or mutated, but they still remain in the genome. They are called pseudogenes here. And you have some gene fragments which again are not functional. You have some mutated genes. So these are the unique sequences. These are the repetitive sequences. And then we have, but these are small sequences which are repeated in the genome. But we may have some, जैसे हमने पिछले slides में देखा, there are thousands of sequences which are repeated, sometimes back to back and sometimes at various positions of the genome. So these genome-wide repeats, they are also non-coding region. And then we have transposons which may keep changing their position, and when they are changing their position, sometimes they fit into a particular gene and then they disrupt that gene. So they also cause mutations. And when we are talking about non-coding, this is the information that we have. Maybe some decades later, when we are talking of the same aspect, these may also, some of these may come into this category. We may know the function of some of these so-called non-coding regions. So we have compared these prokaryotes, eukaryotes with respect to chromosome structure. Okay, structure I have not touched. I will talk them when I'm talking about the prokaryote, E. coli and some viruses. So then chromosome number, definitely here we have few and here we have multiple. Chromosome, as such, the entire genome is present in a region which is not bounded by a nuclear membrane, but it is called nucleoid. So this region where the genome is present in a prokaryote is called nucleoid. This is not bounded by a nuclear membrane, whereas in eukaryotes you have a well-defined nuclear membrane. Genome sizes, density, and all this we have already talked about. Then one thing which I will mention here is that eukaryotes, along with the genome which is present in the nucleus, they also have some organelles which also have their own DNA. And we all know that these are two organelles, chloroplast, present specifically in plants, and mitochondria, which is present in both plants and animals. Okay, these I thought that you should, though I have explained these, but you should ponder on these aspects, because there is a continuous debate over this within the molecular biologist fraternity also, that how does the intron become a part of a genome? Was it that initially they were some genes which became non-functional or became
Your voice is not coming. Please check if you are muted.
since when I'm muted. Am I audible now?
Yes, ma'am, you are audible now. I think
Okay.
from the last
So,
in the last slide you got muted.
okay, okay. So I was talking about this, and I was talking about like in prokaryotes, the genome may be linear or it may be circular. All the genome fractions may be linear, they may be circular, or they may be combination of linear and circular. And we call these genomes or the various chromosomes not by the name chromosome, but generally you have one genome in prokaryotes, and there are some circular extra chromosomal species which we call as plasmids or episomes, and they are different from the normal or mean genome because the organism can sustain itself without the presence of these, because they are extra, you can say they are conferring some extra features to the bacteria or the prokaryote in which they are there. And but if they are removed, they may not be lethal for the sustenance of that organisms. So plasmids are of different types. I'll come to that. Then we have episomes, which are also other extra chromosomal DNA, but these are not like plasmids in the term that, because plasmids are autoregulatory, they can replicate on its own. The episomes, they get integrated into the genome of other organism where there is an infection. It does not form part of the genome, and it is also a non-essential element as the plasmids are. And they are F plus and HFR form depending upon like what is their frequency of combination. So the HFR are the high frequency recombination form because they get integrated into the bacterial chromosome. They can keep jumping in and out of the main genome. Plasmids, they remain generally independent. They are independent with respect to their replication also. They may not wait for the main genome to replicate, that they will not replicate along with it. They may replicate independently and they may have a different cycle of replication. So this is like how the E. coli chromosome look like, a scaffold kind of a structure. But you'll see that definitely they have something holding them back here in these loops which are protruding outside. So this is the organized E. coli chromosome. So we'll see like how these compaction is done. When we are talking of plasmids, they are the ones which can be defined in classified into two different conjugates and non-conjugates. The one which are transmissible are the conjugate ones, and the ones which are non-transmissible, they are non-conjugative. Then we can classify them on the basis of their functions. They are R plasmids, Hall plasmids, F and F dash plasmids. And when we are talking of the compaction, this is how the compaction of a, or you can say the organization of a prokaryotic genome takes place. Here we are specifically taking E. coli, but generally all bacterial circular DNAs, they get compacted like this. That there are some primary level of compaction takes place like this, where some first or primary loops are formed here. And these loops, they are held together with the help of small stretches of RNA. Then comes the second level of compaction, and then individual loops, they are looped on themselves, and they are also held by some proteins. Here they are held by the RNAs, and experimentally this has been proved by scientists that the small stretches of these thread-like structures which are holding these loops here centrally, these are the RNA, they are not the DNA, by use of the enzymes RNAs and DNAs. Because when DNAs will be used, the structure was compact as such, but when the RNAs was used, it opened up. And this opened up because these connections, they were made up of RNA, and RNase is an enzyme which will digest, or you can say which will cleave or denature the RNA. So simple experiments proved that the circular chromosome is folded back 2 times by compacting it by almost hundreds of times. See here you have a 350 micrometers of circular DNA, which is at first level compaction, which is almost 10 times reduced to 30 micrometers. And in another level, another almost 10 times or more than that, it is again compacted to 2 micrometers, and which will look like as it was there in the previous slide. This is the central region, and these are the loops which are protruding out. When we are talking of viral genome is a little bit different from the bacterial genome, because here you may have the genome in the form of DNA or RNA. Again, in the DNA also you have a genome which may be single-stranded DNA, or it can be a double-stranded DNA. Whereas in case of when the genome is RNA, generally we believe it is a single-stranded, but you may have some double-stranded RNA species as well. Examples, there are numerous. You may look into, or you may Google, and when you go into details of individual organisms, you may talk in details about the genomes that way. Generally, when you are studying the virus, you can look into the structures of T4 or some bacteriophages, or the TMV virus. So these are the different viruses which can give a representation of the genome of a virus. But one thing which you should know about the viral genome is that wherever you have RNA as a genome, we classify the virus on the basis of, or the genome on the basis of a positive strand RNA virus or a negative strand RNA virus. So here when we are talking about, see here, if a virus is a positive strand RNA genome, that means the RNA genome directly can be translated to make proteins. So that is a positive strand. But when we say that the virus is having a negative strand RNA genome, that means the host cannot directly, the genome cannot be directly translated to a protein. Rather, this will get converted into the RNA. This RNA is replicated into a positive single strand RNA with the help of some enzymes which we call RNA dependent RNA polymerase, and then that will be translated to form the protein. So on the basis of, though both are single-stranded RNA genomes, both viruses, but one is a negative and the other one is a positive single-strand RNA. So the difference is only that whether it can be directly translated or whether it needs one more intermediate step to get back to that level where proteins can be synthesized. This term you all know, provirus, because abhi corona ka time hai, so you must have read a lot about the viruses, and you must be acquainted yourself with some terms. So we know that like how the viral DNA or how viruses are affecting or infecting individuals. They either the entire virus gets into the host, or sometimes they just inject the genome inside the host cell, where they get integrated into the host genome, acts as a, and this integrated genome is called the provirus. So as the genome gets multiplied, it also multiplies along with that. And once the genome and then they use the host machinery to synthesize some of the proteins which will form the coat or capsid of the virus, and then they burst open the cell, and they enter into the lytic cycle, lysogeny and lytic. These are the different stages of replication, or you can say simply the infection stages of the virus when it is infecting a cell, a host cell. And here we are talking about a bacteriophage, because all those viruses which are infecting a bacteria are called bacteriophages. When we are talking about before entering into the genome organization, we should understand the compare. We can compare the eukaryotes organelle genomes with the prokaryotes, because it is believed that by the theory of endosymbiont, uh, this chloroplast and mitochondria, which were considered to be initially when in during the early phases of evolution, they were independent moieties. They somehow got integrated into a eukaryotic cell, and there, then this with the course of evolution, they started relying on the nuclear genome. They lost some of their own genes, and they later on they became an intrinsic integral part of the eukaryotic cell, and along with the cell they keep multiplying independently or of the nuclear genome also, but for some of the functions of the organelles they are dependent on the nuclear genome. But when we talk specifically of the genome which is present in the DNA of these organelles, chloroplast and mitochondria, we may find extreme similarity between these genomes with the prokaryotic genome. But the See here, no need to get into, I'll just explain you here. When we are talking of two organelles, like what variation they may be having from a normal, or you can say the genome, nuclear genome of an organism. Say for example, mitochondria is there for the respiration process, and chloroplast helps in the photosynthesis process. So the genes which are either helping in the photosynthesis process, they may be the extra things which may be present here in the genome of a chloroplast. And similarly, mitochondria is, you can say, you ascribe the role of ATP synthesis, energy production, to the mitochondrial genome. So anything that helps into electron transport chain generation or synthesis of ATP, all those type of genes, and the basic regulatory genes for mitochondrial structure and chloroplast, they may be present here in the mitochondrial or chloroplast genome. But then many of the proteins which are common to both the general cellular functioning, they definitely take it from the products. Instead of themselves translating them, they are now relying on the nuclear genome. So they are semi-autonomous. They are not completely autonomous. I'm not able to see the comments, but if there is something, if you are not able to understand, you may make a note of that. If you're not able to hear, immediately tell me, please, if I'm not audible. And if the slides somehow are not being shared, sometimes you may tell me.
Kiran ma'am, your screen is visible.
Okay, fine. Because I thought that there was some comment somewhere that it's not visible. Okay, so this is one of the genomes, organelles genome, the chloroplast genome, and you can see that this is circular, as is the case in the prokaryotic genomes, because they are basically descendants of prokaryotes, somehow got trapped into the eukaryotes, and now they are part of it. This is just to give a comparison, like how the huge size of a genome can be compacted into a small cell size. Say, for example, when we are talking about the circular E. coli chromosome, it has a circumference of 1.6 millimeters, whereas the cell size is just 1 by 2 micrometer. So if it is not compacted, organized properly, it cannot be fitted within the E. coli cell. So there is a requirement for organization. Same is the case with the eukaryotes also. Like, say, for example, if a human chromosome is placed end to end, the entire genome is placed end to end, it may stretch 2 meters. But our cells again are microscopic. Though there are variations in the cell size, but they again are within the range of some micrometers. So how to fit that meter long DNA in a cell or a nuclei which is even smaller than a micrometer? So for that, it has to be multiple times compacted so that it can fit into the small cell, and within that small cell, within the smallest part of the nucleus. So you need several levels of compaction. For that, several proteins are required which can tightly pack it up. So we come to this part of organization of genomes. E. coli, I have told you through that diagram, so I'll not go into these slides. When we are talking of prokaryotes, there were several models that were given up or discussed before the actual nucleosome model concept came. So several theories came up and they were discarded. So again, though I have mentioned here, but I'm not going to discuss about these, like what were these. But this is the one which is now accepted. That is the nucleosome model, which was initially given by Kornberg and Thomas. So according to this, DNA is tightly bound to some proteins which are called histone proteins, and they form beads on a string kind of a structure. And what is this beads on a string? I'll show you the diagram. And then it is further compacted into a solenoid structure, and then finally into a scaffold structure, which finally gives you the chromosome. Just before I describe that, I hope you can. This is a thread-like structure. You can see this thread-like structure. This is a highly compacted structure, which will again loop several times to form the chromosome. How to make it play? It's not playing. Okay, I'll just directly go to the YouTube towards the end, and then I'll show you a few videos. Maybe somewhere I have that one. But I'll continue with this. I don't want to break up the continuity here. So I was talking about this, that all huge DNA has to be put in some space which is of few micrometers. So how that is to be done? We need to understand few things, or we should remember few things, that we are talking about compaction of DNA, which is a negatively charged molecule. So how we have to do that? Because compacting it may, because these charges which are there, they may repel each other when we are folding them upon one another. So we need some proteins which may be positive, which may interact with them in such a way that the repulsion is not there and compaction can actually take place. So these are the various stages through which the compaction can happen. This is the simplest form, which we all know that DNA is a double-stranded structure, and I'm not going back into the dimensions here. So this double helical DNA is folded in this thread-like structure. Sorry, this thread-like structure, which I showed you here in this diagram. So it looks like a tight rope which has been interwound several times. So how it is happening here? The DNA is wrapping itself around something which is called the core, nucleosome core, and this is nothing but an assembly of 8 histone proteins. So there are different proteins which are called histone proteins, which are 8 in number. They arrange themselves here in the center, around which the DNA is wound 1/3 approximately. So this forms a first level of organization here, and then the DNA is leaving this structure, and then after a small stretch, which is called the linker DNA, it again wounds itself around the octamer. Octa is eight different proteins which form the core of this nucleosome particle. Okay, so the DNA again 1/3 round here, and then again goes out, and then it again wraps itself around another core of nucleosome. And to maintain these things, because if it is not bound by something, these wounds will open up. So there is another protein which is called which is another type of a histone protein, which is H1 histone protein, which will or you can say seal the entry and the exiting, or the DNA which is entering into the wounding or wrapping here. So this way you have a long stretch of beads on a string kind of a structure. जैसे माला में मोती पिरोए होते हैं, थोड़े थोड़े distance में you can see the thread. Then you have what you call pearl, and then you have again a chain, then you have a pearl. So that is beads on a string kind of a structure. And then this is again, this entire stretch is again folded like this, spirally over one another. So this kind of a coil is made, which will give a second level of compaction. So in the first level here, we are seeing that the dimension is 11 nanometer diameter, and the this much of DNA, which other, if you open up, so 11 nanometers compaction width-wise is done in the first step. And when these strings are again folded back on itself, then it occupies the position of, it makes a thread, or you can say a spiral of 30 nanometers diameter, and it is a long stretch, and the diameter is 30 nanometers. So it is the second level of compaction. Then this entire, you can say, a rod like or a rope like of a structure is again then looped to have a even 10 times tighter structure, and the final dimension or the diameter of this third level of compaction is 300 nanometer. And again, it is just like you have a single thread. उस thread की जो length है पूरी, आप उसको कुछ knots, you take small balls and you make some knots on those balls. So this way now you'll see that the final length of the thread is smaller. Then you again make a small coiled structure of this kind, and you'll find that now the length has even reduced. And then once you have that coil, you again try to fold it over itself like this, makes secondary loops on this, and this way you are even making it even smaller in size. And then there is a final level of these kind of structures which are folded here. They are refolded. See, if you're looking at the diagram, you'll see that how to maintain these loops, but there are several structural maintenance proteins which are holding these loops so that they don't open up. So once they are fixed, you can again roll up them, roll these, either you can arrange spirally or helically to make it even compactor, and you reach up to this level of 1400 nanometer wider chromosome. So this is the final form. So it has to reach to this level, it has to come through various stages. The first stage is the nucleosome. Then you have uh, these rope-like things. When they are folded back here, we call them as solenoid structure. And when these are finally with the help of structural maintenance proteins, they organize a form of a chromosome. Then we call them as a scaffold. So this is a scaffold which results into the chromosome. And this results into a tight, compacted structure of the DNA. But here it is not only, we started with only the DNA, but at every stage of compaction, some proteins are coming and helping them to maintain their structure in the folded and the looped form. So now you have a chromosome, which is the assemblage of DNA along with several proteins, which are structural proteins. They may be histone proteins, which are here, and non-histone proteins. What are the different types of proteins? I don't know why these are not playing up. I'll see to it towards the end. The histone proteins which form this complex, the core of the nucleosome, are of 4 types, which we can categorize as H3, H4, H2. H2 are slightly, there are 2 variants of H2 which we call as H2A and H2B. So in general, there are 4 different types of histone proteins which are present here inside the DNA, or you can say which are forming a ball around which the DNA can wound itself. So we have H2A and H2B. We call it a dimer because they first H2A and H2B, they together interact bond-wise and they form a complex of H2A, H2B. And then you have H3 and H4. So they also come together. Now you have H3, H4, one unit, and there is another H3, H4. These will combine together to form a tetramer. Now the tetramer of H3, H4 along with 2 dimers of H2A, H2B, they will form a histone octamer. And here, because we know that there are charges, all molecules, they have some charges. What is a protein? Protein is made up of amino acids. Amino acids are made up of these different amino acids. We know some of them are positively charged, some of them are negatively charged, some of them are neutral. And we have already talked about that the DNA is negatively charged. So some positively charged amino acids of these proteins will interact with the negatively charged DNA so that they can form a complex and they can be tightly wound around each other. They can form some bonds so that they have this structure is stable. This is how diagrammatically we can show that one of these are the 4 on top and 4 here. 8 total histone core proteins are there around which the DNA is wrapping itself. So it is, and when it is, this is the entry point, this is the exit point, and these the entry and exit points are held together with the help of another histone protein which is called H1 protein. So this becomes one unit of the beads on a string structure. So this is the bead, this is the string. And when it is stretching from one nucleosome to the other nucleosome, we call it as a linker DNA. So this is one nucleosome, this is another nucleosome. And the DNA, now this is a naked DNA. Here it is not associated with any protein. This is called a linker protein. Under microscope, very specialized, good quality microscope, light microscope, you can see some beaded structure. These beads are the octamers, these octamers, or you can say the nucleosome particles. And this is the naked DNA, which is not associated with any protein. When I was talking about the coiling of the beads on a string structure into some coils, so they may associate themselves just like you are just you have a rope and you are just to pack it. So जैसे आप अपना वाटर की पाइप को या किसी को आप एक सर्कुलर उसमें गोल करके आप रैप कर देते हो. So that is the solenoid model. But some of them may not maintain the round circles. वो zigzag कैसे भी करके एक circle बनाया और उसको पैक करके रख देंगे. So this is a zigzag model. How it look like? See, when you are properly arranging them in this circle, okay, so you call this as a solenoid. But when you are not taking care that you are making rounds and you are just coiling the entire rope into different coils, and the coils are just passing a zigzag. So you will, when you look at this, you will find something of this side. So here you see the linker DNA and the octamer or the nucleosome particles. They are present towards the periphery of the circle, but in the zigzag model, it is just haphazard. The linker DNA are transversing across the center of this kind of a cylinder, which will be eventually formed. So these are two proposed models, like how the nucleosome structure is packed to the next level of higher packing. So these are some characteristics of that histones. They are hydrophobic. Some regions in these are acidic. So basically, we talk about at this level only to talk about their interaction with the DNA, which are negatively charged. Besides the negative electrostatic interaction, when we are talking about the organization, like how the chromosomes or how the DNA is associated with the various amino acids or the proteins, there are various types of interactions that are happening. Some hydrogen bonds are formed, some non-polar interactions are happening, some salt bridges and hydrogen bonds. It all depends upon like what is the pH level and how the different ions are integrating with each other, how they are associating with each other. So there are some, these are various names given to these interactions depending upon what type of a bond is formed between the molecules of the DNA and the protein. Besides that, when we talk of a genome, we should also, we generally believe that genome is passed on from one generation to the next generation, and this is heredity. So what is there in our parents, we are inheriting, and we will pass it on to our next generation. But there are few more things which now are being talked about, and that is the field of epigenetics. So here what is happening when we are talking of epigenetics, that means we are talking about some modifications present in the genome which is not there, like which is not gene based. Suppose this is one gene. Okay, this is one protein here. This is H3 protein. So there is some gene which will result into the modification of this protein. Now, modification means this entire protein has some amino acids. These are the amino acid number 2, number 4, ninth position. So these are the various positions of the amino acid. And you can see some MA, all these things written. These are the modifications. M means a methyl group is added to this particular amino acid. A is acetyl group addition. P is phosphorylation, means addition of a phosphate group. So these are different types of groups which may be added to various amino acids, and we say that now this protein is modified. So similarly in a gene also, the gene sequence remains the same, but there may be some changes in the gene, wherein some acetyl group, methyl group, or phosphate groups or ubiquitin, so ribosyl, citrulline, these kind of uh, groups may be added or removed, which may modify your gene. And because of that, the type of protein which is formed, it changes. And this generally happens because of the environmental effect that is modifying your genome. And these modifications are heritable as well. So they may be passed on from one generation to the next generation. Now you may say that uh, it is not only the genetic thing that we are inheriting, we are also inheriting some environmentally induced adaptations done in our previous generation. They may not be genetic, but still we are exposed to those kind of adaptation due to another or the emerging field of epigenetics. Now, getting back again to the compaction level, I was telling you about the loops which are there. The loops are to be held together, otherwise they will open up. There's no point you have a loop made and then again you want to rearrange it, so it may break open. So for that there are some proteins which I was talking about, some structural maintenance proteins. So these are few names. Cohesins are ones, condensins are the other ones. So they are the proteins which help in the formation of the chromosome. So here you have the chromosome structure, and you see that these green things which are wounding around, so these are the sets of the proteins which are laterally and horizontally maintaining the structure of the loops, and cohesins and condensins are these ones which maintain the structure of the chromosome. And when I was talking about epigenetics, the modifications, so normally it is the interaction of the proteins with the DNA. So all proteins, these are the histone proteins. Every protein has a carboxyl terminus, which is C, and an amino terminal. Okay, so you have an amino. All amino acids, they have one amino end and one carboxylic end. So you represent them here as in the form of C and N, and these ends, these protruding tails, the amino terminals, they may interact with some other outside molecules, and they may stabilize or destabilize the structure of the nucleosome. Because see, when I'm saying DNA is compacted, we should understand two things. When something is tightly packed, it is not accessed or accessible by the molecules which help in transcription. So what we say is the gene are not expressed because transcription is not happening. Why transcription is not happening? Because the RNA polymerase cannot go and bind to the promoter sequence because it is hidden here in the loop. Somewhere here, suppose there is a gene, so it must be having its regulatory sequences. The regulatory sequences are completely hidden. So this gene which is present within the coil, it will not express itself. So for that, it has to unfold, it has to open up, it has to release this part where the gene is lying, so that this can be seen by other regulatory proteins which can help the transcription process. So for that, it has to open up. So this compaction and opening up is taken care of by several factors or interactions which are happening between the DNA and other type of molecules which can modify the structure and help in releasing the DNA from this nucleosome structure. So that is very important to understand that it is not always in this beaded form. And from this, we should understand that there are two terms we call as heterochromatin and euchromatin. Euchromatin is the loosened up form of a DNA, or you can say the chromosome. And heterochromatin is tightly packed structure. So whenever a gene is found in a tightly compacted region, it will not be able to, it is not visible to other molecules which can start the transcription process. So we say the gene is not expressing. When it is in the euchromatized region, that means opened up region, then only the proteins which can start or initiate the transcription, they can come and they can start doing their function, and we say that now the gene is expressing. So this compaction and opening up, it is a you can say it's a cyclic process, it is a continuous process. Some regions they remain opened up, the other and the same opened up region may again become compact depending upon the requirement of the cell. If a cell needs the product of this particular gene which is lying here, then it will send some signals to open up this interaction so that this region is exposed. And if the cell does not require this, then the cellular environment will favor the compaction. So these things are all very nicely maintained and regulated. And this is the homeostasis of a cell, how it is maintaining its all internal functioning. I have talked about this. Inactive chromatin means it is tightly packed, compacted. We call this compacted region as heterochromatin. When it is open form, we call it as active chromatin, we call it as euchromatin. And there are certain regions which always remain compacted. So there normally there is no gene. They are the genome structure or chromosome structures which always are tightly packed. Say for example the centromere of a chromosome and the telomeres. They are always compacted. I've talked about this when I was talking about the histone. When histones, the histone proteins, they have some specific groups attached to them. That means this attachment will favor a particular type of function. We call that a particular code is given to this histone if some group is being added here. Say for example, phosphate is added, or a acetyl group is added, or ubiquitin is added somewhere. So different groups, they mean different, their message is different, and that means that there is a different code given to this. So the same histone protein H4, suppose here, if it has these flags, it means something else. If these are removed, that means it is deacetylated, it means different. So the messages are different. So this kind of coding decoding is a secondary level. Till now you must have heard of genetic code, that there are some three triplet bases, which means that this will code for this particular amino acid when translation happens. Similarly, in histones also, some the different amino acids, they may be associated with different uh, groups, you can say, and these attachment of different groups to different amino acids at different positions means something else. So this is a histone code, means attachment or removal of certain groups to some amino acids present in the histone proteins. And they are very important because they help in the uh, you can say the entire function of the chromosome, when it has to open up, when it has to close, when this particular heterochromatization will take place, when this region will be euchromatized, means when it will be opening up. So all these things are very uh, nicely regulated, and they are all controlled as per the requirement of the cell. And we say that the genes are switched on and off. It is not, there's no button which will switch it on and off. These are the molecular interactions between various groups which will make the DNA loosen up or tightly bound to the histone octamer. This is the histone octamer, and here in the diagram you can see that the DNA is touching or attaching itself to the octamer. So it is not a round ball, rather here there are several histone proteins, H2A, H2B, H3 or H4, and these proteins are made up of different amino acids. So here when we are talking of this, there are different amino acids which will be integrating with these parts. Generally, it is said that the minor groove is more prone to bending. You have seen that the core seems to be a little bit spherical. So when the DNA has to wound around this sphere, it has to bend. So the bending is more facilitated towards the minor groove, because the minor groove is rich in AT. The bases adenine and thymine, they are more here in this minor group, which helps in the flexibility so that it can bend and fold the DNA around the histone octamer. So this is another thing. And generally the region away from the histone octamer is the GC rich region or the major groove. So this is again a little bit out of scope, quite high, but still nowadays people are talking about like how the packaging of the interphase chromosome is done, and for that some models are also given. So I will not go deep into these models because I have to cover up one more aspect, that is the DNA isolation also. So just a minute, just have a little bit of time. We have roughly half an hour. Okay, so this is a chromosome territory model in spaghetti. I'll just skip this. This is just again like how the chromatin is wrapped up to form a compacted region, how the chromosomes are present in a particular, see here, by electron, what do you call, fluorescent microscopic. Nowadays people are the molecular biologists, they have come up to this, that all chromosomes, they have a specified bound territory wherein they will be found in a normal interface chromosome, in a nucleus, when the chromosome is in the interface part. So a lot more to be discussed in that, so I'll just eliminate, not go in details of this. This was just because some of you may be interested to know the genome of a coronavirus. So this is just a diagrammatic representation of that. So here internally we have the RNA genome, then we have the outer side envelope. On the envelope we have the spike proteins. These spike proteins can bind to the ACE proteins present in the human cells, and this results into the infection. And once they go inside, you know, like what all things are happening. But see, it is a very small genome, 30, roughly 30 kilobase long. RNA, it's a RNA virus. Now coming to like why we are talking about genome, because we humans are very, you can say, when we are reading about something, we definitely want to know more about that. But why we need to know, want to know about these aspects, is because we want to exploit these facts. And one exploitation industry is the biotechnology industry. So when we are learning about the genome, we want to understand their pathogenesis, we want to understand their genomes, because we want to understand like what happens if something of this kind infects us, like in what way we can use it for our benefit. So now we have the sequences, so we know that various genes, we are utilizing all those aspects for gene therapy and whatnot, for various purpose, transgenics and all. But the first step towards all those, when you go towards the genetic engineering field, uh, from my molecular biology to that, you understand the molecular basis of any living organism, and then you want to utilize those facts to gain some benefit. So there are various branches. Two such branches are genetic engineering, which gives you biotechnology also. So the link here is you work on or you play with the genomes of organisms. For that, you need to have certain protocols by which you can isolate the DNA. I will again cut short the story because the time, it's too much of facts in one single go. See, this is the once you start with the DNA isolation, you this is the result that you will get, and this is how you see a DNA. This is a highly precipitated DNA, and crude DNA that we extract when we are doing extraction in the laboratories. It may be having some of the proteins contaminating this DNA. So there are certain steps by which we have to again purify the DNA and then go ahead with some other aspect. You want to amplify, you do the PCR, you want to know the sequence of the DNA, go for genome sequencing if we have not broken the DNA here. So there are several other steps, but first step to enter into the genetic engineering is to know how to isolate it. So there are some various things that we need while isolating a genomic DNA. Like you need to have some. First thing is that you need a plant material, just like that. Here you have a plant material. We can do that with animals also. Any cell you can take, you have to break the cell membrane open. You have that we do by grinding. Once you grind the thing, we assume that the components of the cell are coming out, because otherwise naturally you just take a leaf and you squeeze it and crush it, so you are damaging the entire cellular component. So when we are doing that, we need to have some homogenizing buffer, a buffer which can maintain a pH condition similar to the intercellular conditions, so that damage is not done. We are isolating, but we don't want to disrupt or damage or break or shear the DNA. So for that, we need some buffers. This is how the leaf or the plant material or the animal cells or tissues are, uh, you can say they are cut shorter, or you have small pieces of that. Then this is one common, because this method which I'm talking about is one where we are using acetyl trimethyl ammonium bromide. We call it as a C-tab extraction method. So here we use this as a buffer here. In this we do the extraction process. So when you have the material, you take, you have, there is a, I am not going into the composition of this because these are all available on the nets. So wherever you write a protocol, you get n number of protocols for your particular system. If you want to, whether you want to work on tomato, or whether you want to work on some marigold or wheat or any other species, for every species you have some protocols, some mix and match you have to do. But for all, there are some common steps that we should understand. And these common steps are that first thing is homogenization, that is extraction, breaking up the cell. Then you have all the cellular components which come into the buffer. After that, then you have to step by step remove the debris, cellular debris, because you are interested into the DNA which is present within the nucleus. So you don't want any of the other organelles, the other cytoplasmic fractions. So you do centrifugation. So we are warming this in the C-tab. Grinding is done. After this ground sample is then put into small test. जैसे हमारे पास test tubes होती हैं, ऐसे ही हमारे labs में when we are doing uh, some experiment in micro quantities, there are some small vials which we call as Effendorf tubes, and these can be fitted into centrifuge machine which are specifically for these sized centrifuge tubes. So the you put your extract into this, and then you are subjecting it to centrifugation. These are various chemicals, all the chemicals that, because you need to break the interaction. Here, see, there are few things which are always there in the extraction mixture. You need to have some high salt concentration. You need to have some chelating agents for that sometimes, and then you have some which can result into the saponification of the cells. uh, the chemicals names are SDS, which is a detergent kind of a thing, which helps in neutralizing the charges of the DNA. You have salt concentration high, which can precipitate the components in the extraction mixture. We have mercaptithanol. Later on, we are using ethanol also for again precipitation purely of the DNA. You have chloroform isoamyl alcohol, which will give you which will separate the various after centrifugation. You have the leaf extract. The extract is done in the buffer or the extraction media which contains all these chemicals, and then you spin it. In the first spin, all the debris, the cellular debris, the heavier metals will settle down in the form of pellet, and you take the supernatant from this, and the supernatant is then to which you add the chloroform isoamyl alcohol. So this will again separate the aqueous phase and the organic phase. Once you get these two layers, you very finely take up the upper layer which contains your DNA, and you put it into another Eppendorf here. So separation, you are just taking one layer which contains the DNA here. The other microsomal parts, different organelles, uh, they are all in the other, either in the first step, if while pelleting, you are removing them, or in the second step, when you are making aqueous phase separate from the organic phase, you are removing all those in the second phase. Then you are just taking the upper layer which contains your DNA, to which you are adding pre-chilled either isopropanol or ethanol, which will precipitate out your DNA in the form of the thread-like structure, which I have shown you in the first or this photograph here. So this finally you are going to get this one. There's a video also for this, but due to the limitation of time, I am not showing that. I am just taking the clippings of all this and just showing you this is the final DNA that you will be getting this. But again, you do not know, because this is a crude extraction. You do not know whether this DNA, how much of this DNA is there. So for that, you need to do some quantification. This quantification can be done with the help of either a agarose gel electrophoresis or spectrophotometrically also we can check for the presence of DNA. So I'll just discuss one part. This is here you have agarose, which is a gel like, when you heat it after mixing it in water, it forms a jelly like thing which solidifies. And here with the help of a comb like this plastic structure, this is called a comb. When you insert here into the gel, you have these small slots made. So here in this, you make a diluted sample of this in TBE buffer. This is again, there is a composition how you go, you are making it, which is available on net. So you make this buffer, you take a little amount of it. Again, a quantity is there in which you have to add your isolated DNA. You make a dilute solution of that. You put it in the slot along with some marker. Again, I do not know whether you know electrophoresis as such or not, because if you are adding here the sample of DNA and then you are connecting it to electricities, uh, the negatively charged DNA will start flowing from the negative terminal of this towards the positive terminal. This is how it looks like. So this system is put in a buffer which is connected to here where you have the positive electrode, negative electrode, and here also you have the terminals. One is the positive, the other one is the negative. The upper side, DNA being negatively charged, it has to move towards the opposite end. So here towards the lower side, we'll be having a positive terminal here. So the DNA moves towards this side. You can see the blue colored structure. Here you add the dye to it, because if DNA is moving, you do not know where it is in the gel. But along with this blue colored line which is moving here in this DNA, I'm talking in a very layman's language. I'm not talking scientifically here, because you will have to have a complete lecture on electrophoresis to make you understand what I'm talking about. So I'm giving you just a glimpse on that this is one method by which. So if it is moving here, then there is a dye called ethidium bromide, which is also added to this, and or you can say in the gel you have ethidium bromide also. So wherever you have the DNA, ethidium bromide is a chemical which gets into the stacks of the DNA. So wherever DNA is, that ethidium bromide also travels along with it. So it will bind to it. So when you expose this gel to a ultraviolet beam, or you put it on an instrument which gives the UV light, so under that a pink colored line will be there somewhere. If the DNA is there, you can just switch on there the UV light, and you can see that a pink color fluorescent band will be there, which can give you an idea whether DNA is there or not. Because only if DNA is there, only then the ethidium bromide will bind to it, otherwise it will not bind. So this will give you an idea that whatever you have isolated, whether it has DNA or not. But then this is not going to give you an idea about how much DNA is there. Because if you want to do some experiment with that isolated DNA, you need to know how much DNA is there in your sample. For that, there are some more experiments by which you can quantify the quantity of DNA present in 1 ml of that particular sample, or 0.1 ml of your sample. So that is again another part of the story. Though this isolation of DNA I just rushed through, but again, just to summarize, our purpose is to reach to the nucleus. For that, you have to break up the nuclear membrane. You have to break up the cell wall, membrane and cell wall. For that, you have to do maceration, or you can say homogenization. That is to be done in some homogenizing solution and the extraction solution. The extraction solution will solubilize all the things that are present in the cell. When you are making a paste of that, so everything which is there in the cell comes out in the chemicals. So with that, you put it into a Eppendorf tube, you are centrifuging. So when you centrifuge, it is just like that. The heavier thing, they settle down, and the lighter thing, they are there. we believe that the heavier things are the debris, the other bigger size cellular organelles, so they settle down. And DNA being lighter, it remains in the suspension, the other, and you can just take the upper part, which is not, and you remove the pellet, take the supernatant, transfer it to another a small Eppendorf tube. And to this, now you gradually keep adding the things which will remove everything in step by step. You keep centrifuging. So there are various protocols which just need 2 centrifugation steps. There are some which requires more. It depends upon like how much pure DNA you want to do, whether it is just to understand the isolation process or whether you want to use that DNA for further experimentation. And in the final step, after separating all the organics and the other contaminants, protein, when you are adding the chloroform isoamyl alcohol, so that is to remove as much of protein which is there in the DNA and in the cellular components. So you want to remove everything. And then you take the layer only, the protein is in the one layer and the DNA is in the different layer. So you take the upper layer in which you have the DNA dissolved, and then you add chilled ethanol to it. The ethanol will precipitate the DNA, which will look like a white fiber or thread-like structure as shown in the picture, and you can just take it out, dissolve it either in distilled water or you can dissolve it in the buffer and store it, and if required you can check it by using electrophoresis method or spectroscopy. That is it from my side. There are these are like the topics which need to be covered in different lectures, but I have tried to again compact these things within these 2 hour slot. I do not know how much you people have actually understood, but still some questions might have come while listening to my talk. If you have any question, you can talk to me, you can ask me. I'll try to answer as much as possible.
किरण एक क्वेश्चन है इसमें
Okay, let me.
किरण बहुत अच्छा एक्सप्लेन किया था. एक स्टूडेंट ने लिखा था मैम हिंदी में भी थोड़ा सा बोलिए.
Okay, I didn't. Okay. I'm
okay,
sorry
no problem.
because while presenting the entire screen is there, chat बीच में मैं देख नहीं पाई. किसने पूछा है ये बात? I'm so sorry. अभी आपको कोई बात समझ में नहीं आई हो, आप कुछ पूछना चाहते हैं तो आप पूछ सकते हैं, मैं हिंदी में बताऊंगी आपको. This is a Simran.
और एक स्टूडेंट इट वाज क्वाइट डिफिकल्ट टॉपिक. तो ये कौन सा क्लास स्टूडेंट है, मतलब
हाँ, मैं इसीलिए मैं बहुत चीजें इसमें थी वो जो मैं स्किप कर गई या मैंने बहुत डिटेल्स में बताया भी नहीं. Realizing that it is going to be going a little bit higher, and I wanted to introduce few topics which are a little bit higher because last time I realized there were many second year students from DU who were participants, और उनका second year में ये genome organization they actually go through. They have an entire unit devoted to that. So I thought that majority of them already have understood this thing, so I wanted to add up something new to that. Okay, so there are few things like somebody is asking, can we call
first semester में भी बच्चे ने पढ़ा होगा.
Okay. Can we call epigenetic genome mutant genome? No, epigenetic genome is not a mutant. Mutation is something. Mutation हम तब कहते हैं जब आपके जो जीन में चेंज आ गया या आपका जीनोम में चेंज आ गया. Say for example, A की जगह G आ गया या T की जगह C आ गया, जिसकी वजह से उसका what do you call, the sequence is changed, and if that sequence is present in a gene, then definitely it will result into a change in the coding. उसकी वजह से प्रोटीन अलग बनेगा, क्योंकि अमीनो एसिड एक अलग कोड हो जाएगा तो प्रोटीन आपका अलग बनेगा. दैट वे म्यूटेशंस आर देयर. और हमेशा ऐसा नहीं है कि कोई म्यूटेशन से प्रोटीन में चेंज ही आएगा. कुछ साइलेंट म्यूटेशंस भी होती हैं, कुछ हो सकता है वो नॉन कोडिंग रीजन में हो, तो वो म्यूटेशन हमें दिखती भी नहीं है. लेकिन ये एपिजेनेटिक्स इज नॉट अ फॉर्म ऑफ म्यूटेशन. दिस इज जस्ट अ मॉडिफिकेशन. your genome remains the same. So as such, we cannot say that mutation has happened. Mutation नहीं हुआ है, आपका genome same वही है. But along with that, some due to some modification to the products of that genome, some extra advantage or maybe disadvantage they happen, which comes under this umbrella of epigenetics. Is operon only present in prokaryotes? If yes, why it is not needed in eukaryotes? Again, specifically we cannot say why it is not needed, but eukaryotes genes are more complex, more cumbersome, and the metabolic or the entire thing that is happening within a eukaryote, there are various, it is a complex organism definitely in comparison to a prokaryote, which are very simpler. So there it can be taken care of that the entire, like for one particular set of a reaction, some enzymes are required and they all are coded by one particular genome. But eukaryotes में जाते जाते जितना बल्कि आपका genome होता चला गया to and same gene may be coding the same enzyme, and the same enzyme may be part of several metabolic functions. So it is not that you can link that to one particular metabolic pathway only. So that is definitely one of the reasons why here we don't find operons. And for every specific gene, you have some. And then because the gene size is too big, you have introns interspersed in between, and for one particular it is you can say expanded genome. Operon concept must have failed initially. हो सकता है lower organisms में हुआ ही होगा, तो evolution में क्या क्या हुआ होगा, I am not in a position to answer that, but definitely to think that a operon system will work as precisely as in prokaryotes, definitely it will not. Then can we use equali? If there are some questions which I am missing out क्योंकि ये chat box मेरा move हो रहा है, तो अगर कोई प्रभा, if you want to answer some of the questions which you feel
है हाँ और एक क्वेश्चन है ड्यूरिंग replication do the nucleosomes also get replicated or they are reformed like other organelles?
क्या है during और okay here I
during
can see the
replication हाँ
during replication, do the nucleosome also get replicated? No, when replication is happening, replication DNA का हो रहा है, तो उस टाइम आपका nucleosome structure को dissociate होना पड़ेगा. तो उस टाइम nucleosome में जो आपके core जो proteins हैं, they are not there, they are removed. क्योंकि आपको बताया ना, जब भी किसी चीज का कोई भी प्रोसेस होना है उसको एक्सपोज होना होगा. डीएनए रेप्लिकेशन के लिए भी आपको डीएनए पॉलीमरेजेस के लिए जगह चाहिए आपको, और बहुत सारी जो प्रोटीन्स उसमें एंजाइम्स हैं जो रेप्लिकेट करती हैं उनको एक्सेस चाहिए डीएनए का. सो दिस इज नाउ ए डिसमेंटल्ड फॉर्म, नाउ इट इज अ लूजेंड अप डीएनए, सो इट इज रेप्लिकेटिंग. पर क्योंकि आपका रेप्लिकेशन हो रहा है, डीएनए जीनोम हो रहा है, उसके बाद वो कॉम्पैक्ट भी होगा, so there are different mechanisms. It is not parallel along with the DNA that nucleosomes are replicating, but the histone proteins are definitely being synthesized more in number, so that now they can also associate with the newly synthesized DNA to make it compact. But it is not like simultaneously एक वो लगा ही हुआ है अभी beads on a string structure है, it opens along with the bead, वो भी half हो रहे हैं और उसके साथ वो duplicate हो रही है, nothing like that. डीएनए रेप्लिकेशन अलग से होगा. जो न्यूक्लियोसोम में प्रोटीन्स रिक्वायर्ड हैं, दे आर सिंथेसाइज्ड बाय सम जीन्स, वो अलग सिंथेसाइज हो रही हैं, एंड व्हेनेवर कॉम्पैक्शन इज हैपनिंग दे कम टुगेदर एंड दे फॉर्म द ऑक्टामर एंड देन दे रैप द डीएनए अराउंड इट. सो दीज आर टू डिफरेंट थिंग्स.
और एक क्वेश्चन है फंक्शंस ऑफ एपिसोम. Some more functions of episome.
Episome is basically, it confers more of you can say because see these are the ones which they can go and integrate in the bacterial, in the genome, they can move out of that. Some of them are highly replicative, but as such, like see जैसे plasmid का एक है, these are you can say जैसे plasmids are the one which can confer the fertility factor or they confer resistance to antibiotic. But episomes are extension of the extra chromosomal genetic material. They definitely confer some selective advantage with respect to some aspect or environmental factor, may not necessarily be antibiotic resistance, but it is conferring some advantage with respect to a changed environment. Adaptation may help करेंगे, but they are not required for the basic essential maintenance of their body. Or you can say survival के लिए it is not required, but it does confer some selective advantages, and different episomes may be conferring different type of advantages, all of which at this moment I may not be in a position to actually tell you like in what all ways they are giving you. Though I know that okay, they have some, what do you call, we have HFR, we have F plus cells. So I don't know. I'm really sorry. I can't comment on that. In what way they are conferring advantages? I haven't gone into that deep. What else I can answer?
एक स्टूडेंट है
Ma'am,
पूछा है बेसिक
can nucleus act on DNA in between two H1 histone, that is linker DNA, because it is free? Nucleus as such DNA, when हम at what point you are saying intracellularly पूछ रहे हैं, तो nucleases अपने सेल्फ डीएनए को कभी क्लीव नहीं करती है, और you are saying H1 is a linker histone, that is linker DNA. No, nucleases will not act on its own DNA. जो अपने सेल में डीएनए है, चाहे वो opened up है, जो linker DNA है, no, it will not.
और एक स्टूडेंट है किरण, ये पूछ रहे थे जीन, so how we can use E. coli if gene have different size? जीन का तो साइज सेम ही होगा, बट जीनोम में भी डिफरेंट, जीनोम साइज में भी डिफरेंट है ना.
How can we use E. coli if genes have different sizes in bacteria and humans? We are using E. coli, we are not using E. coli's gene, we are using E. coli as one of the, you can say, vectors. और vectors are the plasmids, and these plasmids जिनके अंदर हम अपनी genes डालते हैं, उनको propagate करते हैं in some bacteria. So I do not know from where this question, like what exactly you want. You can unmute yourself now and you can ask if you want to. We are using E. coli, but there we are not inserting the gene here in the E. coli genome, or it has nothing to do with the size of a human gene or any organism's gene. There is some misconception here. We are using E. coli just for culturing or propagating the plasmids, which are being used as the vectors in which we are inserting any gene of interest to make a transgenic plant.
So maybe ये different होगा जीनोम
ये क्या कह रहे हैं मैम?
साइज,
यस
जीनोम साइज इन बैक्टीरिया ह्यूमन में डिफरेंट साइज होगा.
हाँ जी बेटा, फिर से बोलिए.
यस मैम, जीनोम के लिए बोल रहा था मैं. एक्चुअली उस वक्त ध्यान नहीं आया तो जीन लिख दिया. एक्चुअली जो आपने जो बताया शुरू में कि प्रोकैरियोट्स या और डिफरेंट वेरिएशन में साइजेस के होते हैं जीनोम, तो हाउ कैन वी यूज डिफरेंट मतलब बैक्टीरिया के लिए in...
No, but now my explanation, has it answered your question, your query या जो doubt था? Here we are not, because we are not taking the entire genome and putting it there in a bacteria. We are just taking a selective gene, we are putting it in a plasmid, and then which is being put into the bacteria. If you are talking about gene cloning, if I'm getting you. आप स्पेसिफिकली जीन क्लोनिंग से रिलेटेड पूछना?
मैम ये बस ये पूछ रहा हूं कि जो डिफरेंट साइजेस है, साइजेस को लेकर अगर डिफरेंट साइजेस हैं तो अगर हम लोग डिफरेंट उसका यूज कर रहे हैं बैक्टीरिया का और ह्यूमन का रिलेट करके, तो उसमें साइजेस क्यों नहीं इफेक्ट डालता, मतलब साइजेस इफेक्ट नहीं
साइज का
डालता?
नहीं, हम वी आर इंटरेस्टेड इन द प्रोडक्ट ऑफ अ पर्टिकुलर जीन. इफ देयर इज सम जीन ऑफ इंटरेस्ट इन अ बैक्टीरिया, अगर उसका हम कोई प्रोडक्ट यूज कर रहे हैं, सो इज दैट व्हाट यू वांट टू नो, और उस प्रोडक्ट को हम ह्यूमंस के लिए यूज कर रहे हैं. I again am not getting this because see the size, we are not in any way putting the we are interested in taking a gene and putting it in some organism so that that product can be formed. But the size of the genes, they do not matter that much when we are putting them. Eventually, ठीक है, bacteria का gene है, that is smaller. We are not putting a human gene in a bacteria, but human gene definitely, wherever you are putting the gene, the size is not that important. You need to have the regulatory sequences wherever you are inserting a gene, कि वो उसको एक्सप्रेस कर सके, उसको ट्रांसक्राइब कर सके, ट्रांसलेट कर सके. तो इसके लिए हम हमेशा रेगुलेटर सीक्वेंसेस, प्रोमोटर्स ऐड करते हैं, क्योंकि इनमें जो मिसमैच है या जो कंपैटिबिलिटी प्रॉब्लम है, वो सिर्फ ये आता है कि प्रोकैरियोटिक प्रोमोटर्स थोड़े से डिफरेंट होंगे, यूकैरियोटिक्स अलग होंगे, यूकैरियोटिक्स में भी प्लांट के प्रोमोटर्स कुछ अलग होंगे, एनिमल्स के प्रोमोटर्स कुछ अलग होंगे. तो जनरली कोई भी जीन है, अगर उसको किसी सही प्रोमोटर के साथ आपने लाइगेट कर दिया या फ्यूज कर दिया और फिर आप उसको इंसर्ट करते हैं, इट एक्सप्रेसेस. इट हैज नथिंग टू डू विद द साइज. एटलीस्ट टिल नाउ आई हैवंट read any such article where the size of a gene, if you want to introduce it somewhere, it has affected the process. Except for that, yes, expression नहीं होता बहुत जगह पे. उसके लिए यू लुक फॉर कि आपको क्या रेगुलेटरी सीक्वेंसेस डालना है, क्या अटैच करना है, जिसकी वजह से उसका एक्सप्रेशन हो सके. Does that answer your question, Ayush? I'm not sure.
Yes, ma'am.
Then there is like adding phosphate group and methylation. You told some different proteins are formed from normal. No, not different proteins are formed. उनका function change हो सकता है. कुछ different groups के डालने से addition of phosphate, methyl, or these groups, when they are added to different proteins, definitely their nature changes. It is not that the protein completely changes. प्रोटीन तो सेम रहता है, बस प्रोटीन का क्योंकि कुछ ग्रुप्स आने की वजह से उनकी जो एफिनिटी है किसी और ग्रुप के साथ कम या ज्यादा हो जाती है और उनका फंक्शनिंग में इफेक्ट होता है. बिकॉज सम ऑफ द प्रोटीन्स, सपोज दे आर एंजाइम्स, डेफिनेटली देन they are acting on some pathway. They may be changing, or they may be metabolizing something. They may be catalyzing some reaction. Maybe they may become inactive or active after addition of some groups. Just an example. And all proteins are not enzymes, I'm telling you, but just to give an explanation, addition of some groups may change the nature, the way a protein can affect a particular reaction. So that is how it is, but it is not going to change the protein as such. Core to remain same. And it is not like what epigenetics की protein change हो गया, that is epigenetics.
यहाँ भी एक एपिजेनेटिक्स का है. एपिजेनेटिक्स चेंजेस अकर इन माइटोकॉन्ड्रियल डीएनए.
Mitochondrial DNA genome में, so wherever the genome is getting some flags or some additions or changes in the groups, addition या removal से है, so it definitely may be affecting. Definitely it may be affecting. And these changes, but the thing is like studies, so we have to example भी देने के लिए I'll have to look into some example which I am not sure of, but as per the basics of epigenetics, it definitely changes. Anywhere it can happen. It is a simple fundamental of addition or modification of group additions to the products which are there in the genome, which are coding for the coded by the mitochondrial genome. So there may be some, because epigenetics, again I'm telling you, is it's not a very new, people are working on it, and we have all examples which state that there are changes which happens by addition or flagging of some of the protein groups, as it happens with addition or modifications of some nucleotide bases. So similarly here, not on the DNA, but on the protein products of the mitochondrial genome, epigenetic changes may happen. Definitely it may happen. How much reliable is spectrophotometric determination of DNA? It is very much reliable, even more because here you can exactly quantify the amount of DNA. Yes, it does require standards. Can we, okay, this is. What's the heading? Yeah, I am sorry for this. What's the heading of this page? Because there was a bar which I was not knowing how to remove that. So wherever like there was the requirement, I read that topic. Say for example, when I was talking about the first time which I realized that there are bars on the headings, I spoke about that. I'm talking about C-value paradox. And the topic heading of all the slide was repeated there in the points also. I'm sorry for that because I couldn't remove that. That must have definitely bothered you all. Any question which I'm mutant? Yes, I understand it was a difficult topic. Even I found that I could not do exact justice to the topic because एक साथ बहुत सारी चीजें बताने की कोशिश में I might have missed the basic. Because to build up a particular topic, when you're talking of epigenetics itself, there is a lot more that has to be explained.
एक तनुज भी लिखा है, it is amazing session. One of the amazing session भी लिखा हुआ है.
I hope it's good that somebody could make out essence of this lecture. I agree to that. It was a difficult one, and it was so. If I am going to give a repeat of this in other programs, definitely I'll just cut short few things, or at least take up few topics and elaborate upon that only. I realized while speaking that this has become a little bit high level. But even if 50% of it you students could understand, that will make it a worthwhile session, because when they look into or read the things, they may remember. At least they'll be familiar with the terms that yes, these are the terms associated with when we are talking about genomes. Anything that is coming up to your mind, you can unmute and you can ask, please. What is that histone DNA interaction? Yes, histone and DNA, they do interact. There are I talked about various bonds also which form between histone and DNA, so that they can associate to form a compact structure. Nothing more. Can we wind up, Anurag?
Yes, ma'am.
Okay, where is it? Okay. So should we end the meeting then?
Yes.
Hello, ma'am.
Yes.
Hello.
Yes.
Ma'am, I have a question. Actually, I asked my question in the chat box, but it is not yet answered. So I wish to ask.
Please. Yeah, please ask.
Ma'am, in between you said that DNA can be quantitative determination can be done by spectrophotometric or spectrometric. So how much reliable is this, and is there any use of standard?
Yeah, I answered this question, Bhupen. I told you that it is very much reliable. Most of the laboratories who are working on the DNA, they do check or estimate the presence, contamination, and the quantity of DNA using spectrophotometric methods. It is quite reliable, and yes, definitely we need to have some standards for that.
Okay, ma'am. Thank you.
There is one more I can see, like how would they calculate how much gene have different various organisms. See, now the genomes of various numerous organisms have been sequenced, and a gene has a specific structure. And now we have everything is, it's not done manually, it is highly computerized and automated. And given a sequence of a particular gene, how many are there in a particular genome, which is fed into the system, the sequencers, automatically you get the answers like how many genes are there. Yes, the genomes, they are sequenced. From there, the annotation, the specific sequences are given, that if these are the regions which are flanked or by these particular sequences, that is one gene. So accordingly, the software takes up that this is a gene, and they tell the number, like how many of these stretches are present in the entire genome. So that way, this is Ayush asking the question.
Okay, ma'am, I think there is no more question.
Yes.
So,
one thing which yes, episome wala bachchon, please एक बार आप लोग भी देख लेना, because right now I am not getting anything in my mind as a function for a particular episome. So for that, definitely you can look into. जिसका भी question था, I say sorry for that. I could not answer that. Okay. Suraj, you want to ask something? Okay, then Anurag, I think we must close
yes,
it because
ma'am.
it's more than 2 hours now.
Yes, ma'am. Great.
Okay.
So participants, thank you for joining, and you can
Yeah,
leave now.
thank you. I must say thank you for your patience. From your
Yes, ma'am.
comment, I realized.
Yes, ma'am. So we would ask the participant to leave, and we can end the meeting. Please leave the meeting.
You can end the meeting, yeah.
Yeah, Anurag, I think host can end the meeting.
Yeah.